And it is now 3 o'clock. So we'll get started. Welcome to the department's 7th virtual colloquium of the fall term. And today we're bringing Stephanie Mundis, who will present the talk of spatial patterns and mosquito characteristics that mediate vector control effectiveness and disease risk in Florida. Now, this is still sort of along with the sub-theme this semester of biologists doing geography and geographers doing biology. But Stephanie actually has degrees in both geography and biology and anthropology. So she's got a leg up on all of us. At any rate, she does have a bachelor's degree at the University of Illinois in Urbana-Champaign in geography and GIS and anthropology. And she has a dual master's degree in biology and geography at New Mexico State. Then we were able to attract her here to work in the quantitative disease ecology and conservation lab, Dr. Ryan supervising, where she's a doctoral candidate now. So that reminds me and reminds all the faculty who are tuned in, and I'll send you the form in a second, please fill out the one-page evaluation form so we can put it into our student learning outcomes files so Jason can write a report about it later on in the year. And all the students now who are taking the class for credit are still required to fill out the longer form to do that. So with all that said, welcome Stephanie, and the floor is yours.
Okay. Okay, so can everyone see my slides? Good. I'll assume yes.
Yes, thank you.
All right, great. So thank you for that introduction. And as Dr. Binford said, I'll be presenting on some of my doctoral dissertation work looking at spatial patterns in our Florida mosquito populations. So just a little bit of background about my own kind of academic upbringing. Like Dr. Binford said, I have studied geography and biology, but more specifically, I tend to draw from methods in landscape ecology. So I'm really interested in how factors such as land cover composition, land cover configuration influence arthropods specifically. And I get even more excited when those are arthropods that happen to transmit horrible diseases. So that's where I see myself as situated in this overlap of landscape ecology. I tend to draw a lot on methods from that field, but then also thinking about medical geography and trying to understand from a spatial perspective how those things influence human vector-borne disease risk. And so this is an area of questioning that I was introduced to during my undergraduate years. I also was able to pursue similar research during my master's program. This is a picture of me sampling mosquitoes out in the desert. And I'm really grateful that I get to continue asking these questions now as a doctoral student in this program. So just a quick overview of my two projects. So the first one is looking at Eastern Equine Encephalitis, which I'll be referring to as EEEV, using a single county in Central Florida as the study area. And then for my second project, it's actually looking at statewide patterns of knockdown resistance, so KDR, in a Florida mosquito population. But before I get into both those projects, I think it's really important to acknowledge my collaborators. So for both of these projects I'll be talking about today, these are data sets that I worked with that took years and many people to gather. So I would not have been able to do this work without these collaborators. So specifically, the folks at Orange County Mosquito Control, I was working with a data set that they've been gathering since 2003. So this is something that they have curated over time, and it's, you know, many, many human hours of work, much more than I could possibly ever do alone. And they were willing to not just share their data, but talk with me about their program and all of the work that they do. Similarly, the folks at the USDA Center for Medical, Agricultural, and Veterinary Entomology, they worked with Florida mosquito control districts across the state to gather data on insecticide resistance. Again, this is something I absolutely could not have done alone. And they were willing to work with me and share their data and also talk with me about all of the steps they did to gather that and process that. I'd also like to thank my committee members, most especially my committee chair, Dr. Sadie Ryan, also Drs. Blackburn, Dr. Mao, and Dr. Kaufman, as well as all of the members of QDEC. So past and present, our current, my fellow PhD students, past and present postdocs, as well as our past master's student. So those are the folks who really made all of this possible and have given me a lot of support and encouragement and feedback over the past few years. So diving into Eastern Equine Encephalitis virus. Okay, so why should you care about EEEV? First of all, this is a deadly neuroinvasive virus that can affect humans and horses. So when I say deadly, the case fatality rate for humans is approximately 30%. And in equine cases, it's I've seen figures as high as 90%. So while there is a vaccine for horses to prevent infection, if a horse is not vaccinated and they get infected, it's a very good chance that they're going to die. And in human cases, for the people who do survive infection, there's a good chance that you'll have long-lasting debilitating sequelae. So this can be things like impaired movement, impaired speech. And so there's really a much heavier burden than just the numbers alone can imply. And this is a virus that's been in the news a little bit recently. So these are two instances in Michigan where there was a human case and that was a fatal case. And there was also an equine case in Michigan. And if you can remember a time before the never-ending year of 2020, EEE was also in the headlines in 2019 because that was a particularly bad year for EEE across the United States. So while on average the cases are down here, we had 38 cases across the United States. And so again, while that doesn't seem like a lot in terms of just the raw number, it is a debilitating disease and it has a really high case fatality rate. So it's really something that people are alarmed about when you see this uptick in transmission. And more specifically, why are we interested in looking at EEE in Florida? So if you look at the map of human cases from 2010 through 2019, you see that there are a couple states up north, so Michigan and Massachusetts have a higher total number of cases, but it's important to note that Florida serves essentially as the source for some of these northern outbreaks. So because of the warm climate and the mosquito populations present here, we can have transmission occurring year-round, including through the winter, and we'll have virus circulating between birds and mosquitoes, and then the migratory birds move up northward and can seed outbreaks there. So there's a lot of interest in understanding EEE in Florida specifically because it's the source of EEE in many other parts of the country. So how does EEE transmission work? At first glance, it seems like a classic case of mosquito meets bird. The problem arises though when an infected mosquito then bites a human or a horse. So both humans and horses are dead in post, and that means that we won't sustain high enough levels of viremia, or our virus won't replicate enough inside of us to then infect the next mosquito that bites us. However, the picture gets even more complicated when we ask which mosquito is biting us and why. So for example, Culiceta melanura is the primary vector species. And this is a species that is usually very ornithophilic, which means that it typically prefers to feed on birds. Sorry, I'm looking at the chat.
Yeah,
Okay.
Stephanie, I think you've got your Zoom controls up. Do you think that you could close them? Or you can hide the Zoom controls if you use the three little dots.
Is that better?
Yes, thank you.
Okay, sorry. Okay, so Culex erraticus shown here with the yellow border is a little bit more of a wild card. So in Bingham et al. 2016, the authors showed that Culex erraticus has a vectorial capacity that's about 44% that of Culiceta melanura, meaning the species isn't as likely to be transmitting EEE, but it is also a species that will shift between avian hosts, so biting from birds, to biting mammals. So that means that this is a vector species that can play an important role as a bridge, where it's connecting the humans to this cycle. Similarly, Cocolatidia perturbans is a mosquito that can transmit EEEV, and it's known to bite a wide range of hosts, including both birds and mammals. And we also have Culex nigripelvis. And so this is a vector species that is really abundant in Florida, as you'll see. And it's another one that will feed on birds, but will shift seasonally from birds to mammals. So this is actually just the short list of suspects. There are actually more vector species that are capable of transmitting EEEV, but these are just ones that were present in any number in the study area I was looking at in Central Florida. We also have to think about the reservoir host species. So Culex melanura has been shown to preferentially feed upon passerine birds, most particularly northern cardinals, while Culex erraticus is often feeding on birds like American robins. And so the adult bird density is important, but another component of this is actually nestling birds. So in a modeling study by Unash et al. In 2006, they actually showed that the relative abundance of nestling birds was really important in driving that springtime transmission. Because birds, when they're in this relatively defenseless state, they don't have proper plumage to protect against mosquito bites, they don't have defensive behaviors, they're really easy prey for mosquitoes, and they also sustain high levels of viremia for longer than adult birds, because they don't develop antibodies as quickly. So they can be an important source of EEEV transmission in the springtime. There are also wading birds that have been implicated as important host species in some locations. So for example, in one study in Louisiana, up to 90% of yellow-crowned night herons were shown to be positive for EEEV and capable of transmitting it to the next mosquito that bites them. And things get even more complicated when we find that reptiles actually can be competent hosts of EEEV, and that these vector species are feeding on reptiles as well. So for example, cottonmouth snakes have been shown to be competent hosts, and they also will sustain viremia for longer, and they'll sustain viremia even when they go into hibernation and out of hibernation. So that means they may play a really important role in that overwintering process of the cycle. Similarly, anoles, when they become really abundant in the springtime, they're also competent hosts, so they can have virus and transmit it to the next mosquito. And a lot of these mosquitoes will shift to biting on anoles when temperatures warm up and these anoles come out of brumation. So there are a lot of moving parts in EEEV transmission here in Florida. And all of this EEEV risk is playing out across space and time. So for example, looking at the landscape, wooded wetlands play a really important role in the development of Culex melanura. They prefer to lay their eggs at the base of these trees in underwater crypts. And the water bodies will tend to sustain populations of the wading birds, for example, that play an important role as a host. And then also when we have pasture land where horses are in close proximity to these wetlands where there is high transmission risk, that's where you have the potential for spillover into horse populations. Sorry. Similarly, the weather patterns can play an important role. So there's this intuitive link between temperature and mosquito-borne disease, where when things get warmer, you'll see more mosquitoes, more mosquito-borne disease. And it is true that the majority of EEEV transmission is occurring in the summer, and there's this contemporaneous association there. But there are also factors such as what's happening the winter before. So in Burkett, Cadena et al. 2012, they actually found that very harsh winters will push back the timing of bird breeding, and then that in turn pushes back the timing of host shifts. So The temperature in the winter 6 months preceding can determine the timing of EEE amplification in the spring and summer. Similarly, oftentimes you might think that more precipitation equals more mosquitoes equals more mosquito-borne disease, but it can be a little bit more complicated than that when we start to think about the timing of precipitation events, the intensity of precipitation events, and how all of that relates to host and vector ecology. And then we have long-term climate patterns. So El Nino and La Nina will have impacts on Florida winters, with El Nino resulting in cooler, wetter winters, and La Nina resulting in warmer or more mild, drier winters. And so those have been shown to influence whether you're going to have EEE transmission the following summer as well. So the study area is in Orange County, which is located here in Florida. You see that the bulk of the center part of the county, this is Orlando, and then you have agricultural land cover over on the east side. So they have 11 sites where they monitor sentinel chicken flocks for arthropod-borne virus antibodies. So this is done by taking a blood draw from the chicken, and then you screen it for antibodies, and if it tests positive, that means that that chicken was fed upon and the virus was transmitted to that chicken. So it's important because then you know the approximate time and the approximate location where you have EEE or other arbovirus positive mosquitoes being active. And so those sentinel flocks are shown in the gray squares. And they also have mosquito, they also have mosquito monitoring sites, which are shown in these black points. So I had 4 research questions going into this project. First, what are the land cover characteristics associated with EEE activity? And then what vector species are present at sites with EEE activity? And those were more of the exploratory analyses, and they fed into the second part, which is looking at developing county-wide and then fine-scale time series models to identify what factors precede EEE detection. Okay, so first looking at land cover. The sentinel chicken sites are shown inside of the map, and they're color-coded and sized according to the number of times EEE was detected at those sites. So we have a range from this site, which was the highest producer, where it was detected almost 60 times. And then just north, you have a site where it was never detected at all. And so the land cover variables I looked at included percent wooded wetland, mean wooded wetland size, percent lake cover, and mean lake size. And because this is clearly a smaller sample size, I used the robust base R package to be able to do some exploratory analyses to see if those variables are associated with an increased count of EEE detection. And all of these variables were extracted from a fine-scale land cover data set from the Florida EPA. And the buffers were a 4-kilometer buffer from the sentinel chicken sites, and that corresponds to the flight range of the primary vector of the disease. So I also was looking at the vector species present. So you can see that at some of these sites, these 4 vector species make up to 75% of the mosquitoes found at this location. So that means that a large portion of the mosquitoes that are active are capable of transmitting EEE. And then when you look at just the vector species and their relative abundance, you see that the site with the most activity is also the one with the highest proportion of Quilosetta melanura, and that some other sites have no activity of Quilosetta melanura at all. So for fitting the county-wide model, first I extracted temperature data and precipitation data from weather stations that were found in and around Orange County. So rather than just focusing on these weather stations inside the county that would be heavily influenced by the urban heat island effect, I pulled from stations that were around the county as well to kind of balance that out. I also downloaded data on the Southern Oscillation Index representing El Nino or La Nina years. And so That was another explanatory variable I considered at this scale. For developing the fine scale models, I used land surface temperature data, which was extracted from the 4-kilometer buffer of the sentinel chicken sites. And this is, I aggregated to the monthly level, so the mean land surface temperature at each sentinel chicken site for 2003 through 2017, so 15 years. And then similarly, the same process, but using normalized difference wetness index. So this is a product also derived from Mundis satellite imagery, measuring liquid water content within vegetation. And I did the same process of creating monthly averages for the study period of 2003 to 2017. So when I first started looking at fitting these models, I was planning on just doing kind of a traditional regression-based linear regression analysis. But it seemed really clear that some of the associations were not linear and were not consistent over different temporal lags. So there is a clear pattern of seasonality where you see more cases or more detections in the summer. But there's also this issue of the association between the explanatory variables and the response variable varying over time. So just looking at temperature, this is showing the association between temperature this month and the likelihood of detecting EEE, and that's a positive association. But when you look back 6 or 7 months, it becomes a negative association. And similarly, if you're just plotting raw temperature and raw EEE counts, it's not quite a linear association. It seems to be more of, there's a thermal threshold where beyond that, you're not going to have transmission occurring. So for parameterizing these models, I first fit a null model with just sine and cosine variables, so that accounts for that seasonality. And then I added predictors using hypothesized lags and functions. So this was based on 3 things. First, looking at all of my exploratory data plots. Secondly, looking at the literature and reviewing papers that have used a similar approach for modeling mosquito-borne disease. And then also just thinking about the biology of all the different interacting components of the system. So for the countywide model, I was able to use a quasi-Poisson regression because there was adequate count data so that I was modeling the number of times EEE was detected in the county. And then for the fine scale data, I use a logistic regression model as the base, and I use the land surface temperature and NDWI as the explanatory variables. So it's important to note here that the lags vary across the variables. So precipitation is likely to be important just for 2 months prior to a month, where something like the Southern Oscillation Index might be influential for up to a year. So looking at the results. So first, in the land cover analysis, I found that EEE detection was positively associated with just % wooded wetland, and that was significant. And things like the patch size of the wooded wetland were not important in determining how often EEE was detected at a site. Looking at the vector species composition at these sites. So first, just looking at what was around when a site was positive in the winter, almost 50% of the mosquitoes were Culiceta melanura. So this makes sense, it's the primary vector, and it's also known to be active throughout the winter. However, in the spring, you have this preponderance of Coquilitidia perturbans. So I definitely wouldn't say that that means that this is the species that's now involved in transmission at these sites. It's much more likely that because Coquilitidia perturbans has this kind of mass emergence in the spring, and this is happening in wetland sites, it's just more likely that that mosquito happens to be booming at a time when EEE, at a time and a place where EEE is occurring. However, this is cause for concern because this is another one of those bridge vectors that will happily bite a bird and then bite a human. So it can be a potential bridge vector scenario. Moving into the summer, you see that there were more Culex erraticus mosquitoes relatively in the positive sites than in the negative sites. So that's showing that, again, you have that instance where there's potential bridging between the zoonotic cycle and the human spillover, the human or horse spillover. And in fact, this is the time period when you do see the most human and horse cases in this study area. And then following up into fall, at this point, Culex nigripalpus does tend to increase in abundance overall, and we did see more at the positive sites. So this could mean that at this point, Culex nigripalpus is kind of taking up that baton of transmission and carrying it forward into the early winter. So exploring the county level variables that were used in the county level model. So across these next three graphs, the black line represents the explanatory variable, and the red bars represent the monthly count of EEE. So this is looking at temperature, and we see that there are pretty high peaks that often coincide with the peaks in temperature. Again, we have that summertime transmission. Looking at precipitation, there's some coinciding between precipitation peaks and EEE activity, or the number of EEE detections, but it's really not a perfect fit, and that's why it's important to kind of get in there with the models and try to understand what that association is. And then finally, thinking about the Southern Oscillation Index. So when you have these high values, that means you're having a La Nina episode, and when you have low values, you're having an El Nino. And some of the previous work that's been done at the regional or the state level has shown that El Nino is associated with an increased risk of transmission the following summer. So we did see that in some instances. So for example, December 2004 was followed by one of the higher peaks of EEE activity. And then La Nina is thought to be associated with an increased risk of wintertime transmission. So there are a couple instances where that does line up. So we have La Nina occurring here, and we also had wintertime transmission again here in 2011. But there are some clear parts where it just doesn't match up, and it's not really a clear-cut association, at least from just looking at the explanatory variables. So this is the output from the county-wide model, and it's clearly a lot to just take in, so I'm going to go over it a little bit step by step. So first, this is showing the association between maximum temperature here on the left-hand panel and the relative risk of EEE detection at a temporal lag of 0 to 2 months. So if you have high temperatures in the county that month, you're also more likely to have EEE activity in the county that month. Similarly, if you're at average temperature levels, you're going to have a slightly elevated risk. And if you're at lower county level temperatures, you're going to have a lower risk. So this makes sense. Like I said, a lot of EEE transmission is occurring in the summer. When we look at precipitation, I was actually surprised to see that maximum precipitation can decrease the risk of EEE detection for up to 2 months. So this was surprising to me, but then thinking about what it means to have maximum precipitation in Orange County or Florida more broadly. So this is when I was in Orange County myself, and this was a rain event that was so severe, it was, flipping palm trees inside out like umbrellas. And so it seems to make sense that that could kind of knock down mosquito populations and disrupt virus amplification. We also saw that a period of very low precipitation was followed by increased risk about 5 months later. And this is actually similar to what's been shown with St. Louis encephalitis virus in Florida, where a period of drought seems to trigger a later period of virus transmission. Similarly, for La Nina, it seemed to have this slightly increased wintertime risk. So La Nina and El Nino events tend to be temporally autocorrelated. So if you have a La Nina this year, you might have higher risk of wintertime transmission the next year. And then for El Nino, it did seem to result in this elevated summer detection risk. So finally, looking at the fine scale model, land surface temperature and NDWI. So just looking at the initial variables, I was interested to see that at the site with the most EEE detections, so the most frequent detection, that was consistently the coolest. So in this graph, it's shown in dark red, and it's almost always the coolest. You also can see that there's a wide range of temperature variation during the summer periods. So this cooler site might be almost 10 degrees cooler than the really hot sites. And generally the hot sites were not as active for EEE. For the NDWI was actually the inverse was true. The most productive sites were generally the wet sites, which makes sense. And then in looking at the outputs from the fine scale models, it might just be that the hottest sites are too hot for EEE. So a lot of work has been done, a lot of that work by my own advisor, looking at thermal limits. So it could just be at those highly urbanized sites where the temperatures are above the maximum threshold for virus development in the mosquito. You're just not seeing transmission in those locations because it's too warm. And those actually fell outside of the thermal limits for EEE for most of the early spring to early fall season. And then we actually might see that cold local temperature is facilitating winter transmission. And so then we also saw that local dry conditions can lead to an increased risk 2 to 3 months later. So similar to what I saw with the county level model, when you have minimum precipitation, there seems to be this drought process that triggers later risk at a temporal lag. So to conclude on this project, wooded wetlands, but not patch sites, were important in determining how often EEE was detected. So that might mean that it doesn't take a large fragment of wooded wetland to sustain this transmission cycle. The Culiseta melanura is dominant at winter EEE sites. However, other species do become more abundant in the spring, summer, and fall, and they likely are playing that important role in being a bridge vector. Also, there were different associations between environmental drivers and EEE risk when you're exploring things across different times and scales. So that really illuminated to me that it's important to think about how these relationships will vary across different temporal lags. And then finally, the urban heat island effect may actually reduce EEE transmission at some of these sites where it's just too warm for that incubation to occur. So some of the outstanding questions, I'd be interested to see how host community composition changes over time at these sites, and how does sentinel chicken detection relate to human risk. So during this study period, there were about 15 instances of horse cases, and a lot of those were occurring, or all of those were occurring when there was sentinel chicken activity, but then there's often sentinel chicken activity in times when there aren't horse cases. So it must be some kind of human behavior that, that drives that risk to the point of spillover. So for my second chapter, I'm looking at spatial patterns in KDR in Florida mosquitoes. So which Florida mosquitoes? Aedes aegypti, you may be familiar with from various other talks. This is a mosquito that is somewhat infamous as the vector of yellow fever, dengue, chikungunya, and Zika. These are all viruses that, unlike EEE, they can be sustained in a human to mosquito to human transmission cycle. So they don't just spill over, they can be transmitted purely in urban settings. And part of what makes Aedes aegypti so well adapted to being a vector of these viruses is that it likes to live in close association with humans. So these are kind of prime candidates for breeding for Aedes aegypti. And it also prefers to take its blood meals from humans. So it's really well adapted to being a vector of disease for us. So how do you deal with a vector species like Aedes aegypti? So one of the most commonly used tools that are utilized here in Florida and all around the world are pyrethroid insecticides. So the way pyrethroids work is this is, for example, an ultra low volume spraying from a truck in Citrus County, and this is a mosquito's neuron. And so when the mosquito gets exposed to the pyrethroid essentially serves to jam open these voltage gated sodium channels. And once that happens, then you get positively charged sodium flooding the channel, And then you get some mosquito twitching and eventually mosquito death. And so while that's unfortunate for the mosquito, for people who are trying to prevent mosquito-borne disease, it's really the ideal scenario. However, when you treat mosquito populations repeatedly with the same class of insecticide, you're going to select for the mosquitoes that are resistant. So those are the ones that will be able to continue to persist and they won't be susceptible any longer. And when this happens, this is known as knockdown resistance for pyrethroids. It's really problematic because, as you can see in this map, the vast majority of Florida counties use pyrethroids. And it's sort of the #1 tool in the toolkit for when we have situations like this where Zika virus was introduced into Miami. So this can be monitored by doing some kinds of bottle bioassays where you see how many mosquitoes die after exposure. But also there has been a lot of work to identify the genetic underpinnings of resistance in Aedes aegypti. So specifically, there are these 2 genes that are associated with resistance. So there's the 1534 and there's the 1016. And for both of these genes, there are 3 potential variants. So you have your wild type that is the original form, so it's susceptible at both sites. And then you have mutations that can occur all the way up until you have this highly resistant variant. And so this is a mosquito that is mutated at both locations. And this is a mosquito where when you try to do an assay to assess for resistance, it just keeps flying. It is not impressed by any dose of the pyrethroids. So this is a problem mosquito. And so looking at KDR in Florida, so this was data that was collected between 2016 to 2017 at 62 locations across the state. And just at a glance, you can see that there is an alarming amount of red. So again, that's that highly resistant strain. And so when you dive down even further, you can see that there's a lot of variation though across space. So for example, looking at Miami-Dade County, there's sites where more than 75% of the mosquitoes were that highly resistant strain. But there are also just sites a few kilometers away that are only 25%. So this sparked a lot of questions for me. So I wanted to understand what the spatial scale of clustering was for that highly resistant genotype of Aedes aegypti. I also wanted to identify where there are clusters of resistant and susceptible strains. So it's important to know where resistance is, but it's also informative to ask why are there susceptible strains persisting in this environment of intense insecticide use. And then finally, I wanted to look at what environmental factors are associated with the development of resistance. So for the methods for this project, I first used a dual Ripley's K function to assess for clustering in the resistance genotype frequencies across sites. So I ran this across 30 distance bands of 5 kilometers each. I also identified clusters using a multinomial StatScan approach, adjusting for more likely clusters. And this basically builds concentric circles around each site and then compares the probability of finding a particular version within that circle compared to outside of that circle. Finally, in thinking about the environmental factors that might influence resistance. So clearly the actual insecticide applications themselves may be important or are important, but also you're going to have land cover factors like dense vegetation or the distance from the roads where they're doing the spraying, or the proximity of agriculture where they're using the same class of insecticides for treating their crops. All of these factors may also be influencing how resistance is developing over space. And so to represent all of these factors, I imported data sets from a range of sources, including land cover, cropland, different vegetation indices, and different human population characteristics, as well as the reported insecticide use by each district across the state. So all of these spatial data were projected, resampled, and then I standardized the extracted values, and I removed really redundant variables using variance inflation factors. I then performed a model selecting process using the MUMIN package. So the explanatory data layers, and then the response variable was the frequency of this IICC highly resistant genotype. And I ranked the models by the EEEV information criterion. And once I had those top candidate models, I also tested to see if a spatial lag or a spatial error term would improve the fit or the predictive power of the model. So the findings from the dual Ripley's K-plot is that maximum clustering in IICC frequencies is occurring at about 30 kilometers. So that's saying that you're more likely to be similar to your neighbor at a distance of about 30 kilometers, but once you're more than 100 kilometers away, it's really unlikely that there will be any kind of similarity. So this means that you really shouldn't be making guesses about resistance status at one county based on the neighboring county or based on a site that's a couple hours away. Some of the clusters identified, so the highly resistant clusters were usually focal, so they're pretty small sites where you're seeing more than you would expect. And they were all in urban areas along the coast. There are also a couple instances of the more susceptible strain. So for example, here in the Florida Keys, you're more likely to find that more susceptible genotype there. And then looking at the environmental factors. So to walk through this diagram, the size of the box is sized according to the number of times that variable came up in the best fitting model set. So for example, pyrethroid use was always important. And then much to my surprise, pyrethroid use was also negatively associated with the development of KDR in these populations. So one potential explanation I could see for that is that pyrethroid use is just looking at pyrethroids used by the control districts, and a large number of households also use pyrethroids. So it's a really common household product. You probably have it sitting under your kitchen sink. So that kind of local selection pressure could be influential. And then also if a county is using a lot of pyrethroids, it's also more likely to be a well-funded county that's also doing vector control, monitoring for resistance. They may also be rotating their insecticide classes. There are a lot of things that they could be doing that are kind of countering the development of resistance. The other factors that occurred really regularly were all vegetation indices that were positively associated with the development of KDR. So then when I considered the incorporation of a spatial lag or a spatial error term, it turned out that the best fitting model was the spatial lag version that had vegetation indices, percent tree cover, non-tree vegetation, pyrethroid use, and distance from roads. And so this was a model that had an R² of about 0.49, and it actually had the lowest AIC value too. So that means that it had boosted explanatory power without becoming overly complex. So in conclusion for this project, KDR genotype is widespread, unfortunately, in Florida, and this is also true in areas all around the world. There were significant clusters of low, intermediate, and high resistance genotypes. So it's important to note that this is not a static measure where this is your resistance status for the next however many years. Resistance can actually shift and change over time. And so it would be really important to continue following up and seeing if those sites with intermediate levels of resistance are actually just sites in transmission, where they might be becoming more resistant as time goes by. And so in response to the development of resistance in these mosquitoes, you could try to turn to alternative measures of control. So Florida Keys Mosquito Control does some source reduction efforts. But it's important to note that these are the kinds of activities that are really cost and human power intensive. So it's important to try to maintain susceptibility and predict where you can manage mosquitoes with these insecticides as much as possible. So also the landscape factors were potentially drivers or at least influential in the patterns we saw, and that by incorporating that spatial lag term, it did increase the explanatory power of the model. So that can be an important factor to consider. There seems to be similarity between sites, and we can use spatial autocorrelation to our advantage. So to conclude, I have found that geographic thinking can really shed light on the ecology of vectors and vector-borne disease. And so not just, the cool technology side of using GIS or processing hundreds of rasters, but also thinking about how things vary across scales and thinking about place-specific context. All of that really informs my thinking in these kinds of projects. I also think that spatial analysis and spatial methods can play a role in anticipating and responding to mosquito-borne disease. But something that I know I need to continue working at is taking steps to ensure usability. So while it's great to run a million models and identify all the clusters, if that's not something that can actually be used by the folks who are doing the on-the-ground mosquito control and intervention when viruses do get introduced, it's kind of just happening in this academic vacuum. So a lot of the researchers I really admire take those steps to make sure that the outputs from these projects can be used by the people who need it most. So again, I'd like to acknowledge the folks who have been willing to collaborate with me, as well as my excellent committee and my fellow lab mates. And I will happily take any questions.
Thank you, Stephanie. We have about 5 minutes for questions in the official time, and I'm sure that people might want to stick around after the 3:50 time limit's up, as some of you may have to leave. So any questions out there?
Mike, can I start?
Of course.
All right. First of all, Stephanie, thank you. It was a very good talk. I really enjoyed it. I thought it was very thorough. A couple of things popped up into my mind as you were talking. And surprisingly, this one was not about ENSO. You talked about hot and cold as being important there. But previously you'd pointed out that probably the single largest controlling variable in the Sentinel sites in Orlando or Orange County was the wooded wetlands. You're looking there almost at some sort of collinearity. You're looking at the same things, really, aren't you? Is there some issue there that, I mean, if you've got a lot of woodlands, it tends to be a little bit cooler at that that area? Are we looking at the same thing?
I think that is true, and that's probably driving why the highly productive EEE site is cooler and wetter, because it has a lot of wetlands. And for me, it's about trying to untangle the extent to which the temperature contributes and the landscape contributes and the moisture content contributes. But yeah, I definitely think that there's like a clear collinearity. How hot can you possibly get if a good chunk of your Sentinel site is underwater most of the year? But I haven't really found a good way to disentangle that and understand how those variables either additively or interact to contribute to risk over space and time.
Okay, and an observation which I think there was a graph where you were showing the monthly temperatures or monthly precipitation for Orlando for Orange County. Yeah, you got your little black box up there again. There we go. So you're telling me, and I'm being nasty here, you're telling me that you got 400 centimeters or close to 400 centimeters of rain
over the course of a month?
Yeah.
Yes.
Okay. I got 2.5 centimeters in an inch, right?
Yes.
So if I got 100 centimeters, I got 40 inches of rain in a month.
Okay.
That's 4 meters of rain.
Oh, so that is the sum over the 9 stations.
Oh, so that's a cumulative. All right. So really, you might want to, if it's over 9, you might want to divide through by 9.
Okay.
I mean, I just thought, I looked at that, I thought, wow,
Yeah,
that's really hammering down.
that's a good point.
All
Yeah.
right. And while we're and again, just so I don't completely hog it, while we're talking about very large quantities of rain, I have a good friend and colleague in Colombia, Herman Poveda, who's done some work on the incidence of malaria and ENSO. And he actually has come up with the same sort of conclusions that you have, which is that, in fact, when it rips down with rain, which it does there in the La Nina year, it actually, the number of malaria cases go down because it flushes everything, all the containers over top, and it goes, right? But when it's dry, everybody's trying to save water in open containers, and the rivers dry up and they form puddles and disconnected swamps, which are ideal locations. So yeah, indeed, that, you know, if you like, I can certainly send you some of Herman's work as well. Yeah. Thanks, folks. Stephanie.
All right, it is 3:50, which means that the people who need to leave may leave, but I'm sure, Stephanie, you're willing to stick around for a few more minutes and answer questions, aren't you? Okay. Well, I have a real quick question. Your advisor is famous for showing that the assumed temperature and tolerance curves are not Gaussian in some cases. Yeah, you showed a series of nice bell-shaped curves for high temperature, low temperature, and the high in the middle. Are those done from lab experiments, or are those an assumed temperature tolerances?
This one?
No, these are the ones that you had several different species and their temperature dependence, their tolerance to temperature and their optimum in the center. Yeah, that one.
Yeah. So that was a Bayesian modeling, and it was parameterized by lab studies and kind of a wide variety of sources. But yeah, it seems like EEE really similarly has a hard thermal limit. And so looking at the kind of fine scale temperatures, it's about 30 degrees. So a lot of the sites were just going to be too hot for the extrinsic incubation period for the extrinsic incubation of EEE to be occurring in the mosquitoes.
Yeah, I remember a few years ago, Dr. Ryan got in a big controversy because she had some colleagues that were showing that there was a truncated distribution. They just dropped off at some temperature, so it's going to get hotter. So that reminded me of that old story. Anybody else have any comments or questions? Okay, well, I agree with Dr. Whalen. Very well done, Stephanie. That was a nice time to spend and learn more about this. And once again, you are doing biology in a geography department, and you're a geographer doing biology. So no, the theme seems to be holding up even with the graduate students. Okay, so next week we'll have Stephanie Powell, Dr. Stephanie Powell from Florida State University Department of Geography, who is a geographer doing biology. She's actually a macrosystems biology awardee and has some interesting remote sensing approaches to doing things. But please remind everybody, faculty members, please fill out the evaluation forms and send them to me. And then I'll compile them and send them on to Stephanie. But I'll also go into the database for the student learning outcome data. And students taking the class, don't forget to fill out your forms and upload them in the Canvas course. So with that said, I think that, I'll stick around for another minute or two to see before we close down the Zoom meeting. And if anybody else has questions, ask away.